Hey everyone, so this is our second lecture on internal regulation for this week, and we're going to be talking about thirst and hunger for this lecture. All right, so let's start with thirst. So osmotic thirst, there's basically different types of thirst. We've got osmotic thirst, hypovolemic thirst, and then we've also got this idea of sodium-specific hunger. So we are going to start with osmotic thirst. And in order to understand osmotic thirst, you kind of have to understand osmotic pressure as well. So osmotic thirst is basically the thirst we experience when we eat something that's maybe salty or we've had too much salt in a meal or something. So our body kind of triggers this thirst within us. And the reason that happens is because we have this osmotic pressure from our cells. So if you think back to our lectures on cellular structure, where you can kind of maybe visualize that we've got our cell, and then we've got the intracellular space, which is the space in the cell, and then the extracellular space, which is the space outside of the cell. And so when we eat something that has a lot of sodium, basically it puts that balance of around the cell, the extracellular space. It basically kind of like disturbs the levels of sodium, and our cells sort of recognize, hey, there's all these sodium solutes around, and so it basically impacts the cell because the osmotic pressure occurs when solutes are more concentrated on one side of the membrane than the other. So when you eat something salty, those sodium ions spread around that extracellular fluid. And so the result is that higher concentration in the extracellular space. So what happens is certain neurons are going to detect the loss of water that may happen because the osmotic pressure is going to result in drawing water from the cells into the extracellular fluid. So the cells are saying, hey, there's all this sodium, so it's out of balance. And so they basically draw some water from the cells into the extracellular fluid. And so that cues these neurons that would basically be triggered to recognize that, hey, we've got a disruption in our sodium balance here, and we need more water. So it triggers that thirst. So it's basically the osmotic pressure triggering different neurons to detect that loss of water from the osmotic pressure, and then it triggers that thirst and that motivation or drive to drink some fluids or some water. So that's osmotic thirst. And it's different than hypovolemic thirst, because hypovolemic thirst is related to our body actually having depleted levels of different fluids. So for example, if somebody's heavily bleeding or sweating, our body's going to recognize that there's a disruption in the levels of fluids going on. And so it's going to trigger thirst because our body definitely needs it in that moment. And then we've also got this concept of sodium-specific hunger. And what that is that an animal may become deficient in sodium. And so it then basically craves sodium, salty things. So even maybe something that was super salty that someone might not normally want or like, when somebody is sodium deficient, they will actually be craving or be looking for that drink or food that has high sodium levels, so that that deficiency can be corrected. So I really liked this figure that I found that basically shows sort of a map of how this concept of thirst is working, or the mechanism of thirst. So we've got decreased volume of extracellular fluid, or an increased osmolality of extracellular fluid. And so both of these, whether it's decreased volumes of fluid or that osmotic pressure, it's going to trigger these different things. So in this case, if we think about eating the salty food, it's going to decrease saliva secretion. We're going to get a dry mouth, and then we're going to be thirsty. And so ideally, we're going to have access to water and drink. And then when we lose fluids, we're going to have stimulation of osmoreceptors in the hypothalamic thirst sensor. Shouldn't be any surprise that the hypothalamus is involved here. And so again, that's going to give us that sensation to drink. We look for water and then we would drink. And our gastrointestinal tract is going to absorb the water that we need. And that is going to in turn decrease the osmolality of the extracellular fluid, and then increase the volumes of our extracellular fluid. And again, it just kind of is a cycle, but keeping that, it kind of goes back to that term homeostasis. It's kind of our body's trying to keep everything in balance with our fluids and the different nutrients and things that we have in our blood and our fluids. Okay, so what's really going on with thirst as far as biological mechanisms? So we've got some hormones that are related here. Vasopressin is one of the hormones that is related to thirst. So vasopressin actually raises blood pressure by constricting blood vessels that will also stimulate thirst. And increased pressure helps with decreased volume. And then we've also got this antidiuretic hormone, which enable the kidneys to reabsorb water from urine and makes urine more concentrated. So these are our two hormones that are associated with thirst. And something I found interesting is, vasopressin is actually increased when we're sleeping. So, to get us through the night where we don't have to keep using the restroom, the vasopressin is increased. But that is also, it kind of also explains why we become thirsty right before bedtime and stuff, is because vasopressin also stimulates thirst. So that's just kind of an interesting fact to know. And then we also have these different brain structures that are associated with thirst as well. So these first two, the OVLT and the SFO, I'll just let you go ahead and Google on how to say this one, because it's a very long term. And actually, even our textbook said OVLT first and then put this in parentheses. So these two structures are actually around the third ventricle, and they detect osmotic pressure. So that concept we talked about earlier about osmotic pressure, so these two brain structures are the ones sort of detecting what's going on with our osmotic pressure. The SFO in particular has basically kind of like a population of neurons that are going to detect what's going on and increase thirst, but it also has a population that detects what's going on with osmotic pressure and would suppress thirst. So it's kind of, it's basically helping with both to increase thirst, but also to suppress it when we don't need to drink water or fluids. And those axons combine with input from the OVLT, which is also related to basically kind of like our intestinal tract, the stomach, and elsewhere, to provide info and input into the hypothalamus. Then we've got the lateral preoptic area and the surrounding parts of the hypothalamus to control our actual drinking. And the supraoptic nucleus is actually, and the paraventricular nucleus, PVN, those are actually helping to control the rate at which our posterior pituitary is releasing vasopressin. So these two are basically sort of helping regulate that vasopressin, which comes back to that hormone, which is being released by the pituitary gland, or in this case, the posterior pituitary. So I'm a visual person, so I normally like to show some kind of figure or diagram to help put it all together. So here's these different parts we're talking about. We've got the third ventricle, the OVLT, and the subfornical organ. And again, this is in your book, so you can look at these figures and study them to kind of get an awareness of basically where these places are in the brain that we're talking about here. And I found these figures to be pretty helpful. I like to look at flow charts because it kind of helps put everything in in a step-by-step process. So this is just basically kind of like a flow chart to show how our body responds to hypovolemia. So when we have that low blood volume, or our body realizes that our fluids are low, our kidneys release renin. And then what happens to that when the renin is released into the blood, our body is then triggered that basically proteins in the blood, it triggers proteins in the blood to form angiotensin 1. And then angiotensin 1 is converted into angiotensin 2. And then that is what constricts blood vessels and stimulates cells in the subfornical organ to increase drinking. So basically, for hypovolemic thirst, it's a slightly different mechanism in the sense that it's actually going to be triggering that renin and including the kidneys in the process to really try to signal the body that, hey, we need these fluids to be replenished. Okay. So the thirst that we were describing earlier with the OVLT, the subfornical organ and digestive tract, that is more of the mechanism associated with osmotic thirst. And let me just kind of blow this up. I sort of verbally went over this, but let me blow this one up here. So the osmotic thirst is what I was just describing earlier. But hypovolemic is a little bit different in the sense that it's even going to signal the kidneys and blood vessels to let them know, hey, we've got a low blood volume here, so we need to get this corrected stat. And if you've ever been in the hospital or known anybody in the hospital, a lot of times the bag that they hang on the IV has some kind of solutes in it because that's what the body is needing. So water containing solutes. And that's a lot of times when you see that IV drip, it has solutes in it. Okay, so that's thirst. And now let's talk about hunger. So to talk about hunger, it's helpful to have just a general idea of what's going on as far as our digestion, our digestive system. So when you really think about it, our digestive process really starts taking place in the mouth. We chew our food and we've got saliva and things to start breaking down our food already as soon as we put it in our mouth. And then it moves down from there to the esophagus, down to the stomach, small intestine, large intestine, and then we excrete things that we don't need. Okay, and along the way, there's different pieces there. You know, we've got sphincters, which are kind of like these muscles that sort of stay closed until they release to allow food to come in or go through. But here's a visual of that. So we start the digestive process right away as soon as we start eating. Our food comes down here to the esophagus until it's ready to be released into the stomach, in which it starts to break down even further with different acids in the stomach. And then it goes down until we get down to the small intestine. Here's the duodenum here, which we'll talk about in a little more detail in just a moment, but there's a visual of where it is. And then we've got the small intestines, and then going all the way down, you can see it kind of just snakes around. And if you were to ever just kind of roll out the intestines of a cadaver body or something, you would see that the intestines are long. And so we've got this long stretch of intestines until we get to the large intestine, and then it basically finally becomes released by the rectum, and then we excrete anything we didn't use in our body. So let's talk real quick about dairy consumption. So us as humans and other mammals too, when we're born, you know, we're kind of born with this, you know, as a newborn, we're basically, most of us are living off of milk of some form, our mother's milk, or now us as human babies, we might be formula fed, or basically we some sort of milk is what we're drinking and how we're getting our nourishment. So this type of milk, you know, milk in general, you know, has this type of sugar called lactose that needs a certain type of enzyme to process, which is lactase. So when we're born, we have lactase, unless we're a lactose intolerant baby. We have this enzyme lactase to help us break down lactose that we need to drink milk. And so a lot of animals over time, and even a lot of humans, sort of lose that lactase after a while because they're sort of weaned off of milk and encouraged to start eating other foods. So as we grow up, if we don't continue drinking milk, then we don't need lactase anymore because we're not consuming lactose. So I thought this figure was pretty interesting because it kind of showed us that around the world, if somebody lives somewhere where there's just people just aren't really drinking milk, and maybe there's not dairy farms and cows around and stuff, then you can see here like percentage of people who are lactose tolerant, it really goes down, in places where you may not necessarily be seeing people who would be drinking milk. So it's just basically kind of showing that part of us as human beings who drink milk as adults, it's also sort of an environmental thing too, because from a survival standpoint, a lot of animals are weaned off of milk and start eating other types of food. So here's that figure sort of blown up, kind of showing that the lighter areas are places where it's the lower percent of people who are even able to digest milk, and then basically they have an intolerance in those areas. So what factors do we have related to feeding? Our feeding schedules, our feeding regulation. Well, there's definitely oral factors. So our textbook kind of describes how there's even been some evidence found that, back in the day, people had chewing gum, meaning that they were chewing on some bark. So there's an oral factor, meaning that like we have this desire to chew on things and put things in our mouth. And some studies related to sham feeding, where basically people were fed, or really animals were fed, but really it wasn't going to the digestive tract. They were not satiated. So what that told us is it's more than just oral. So it's not just only related to oral factors, but there's also a stomach and intestinal factor there as well. So different research has basically helped us understand that it can be related to our stomach distension. So when our stomach becomes distended, our brain is cued that we're full, but also at the same time, the duodenum, which I had highlighted earlier on that figure of the digestive tract, there's also some different receptors there that also, when that duodenum is full, it also kind of triggers our brain to know that hey, the duodenum is full, and so basically we may not need another meal. So the duodenum is, again, part of the small intestine that adjoins the stomach, and it absorbs nutrients, and nerves inform the brain about the distension of the duodenum and the amount of nutrition. So kind of the nutrition that we need. And it releases a hormone called cholecytokinin. And I may have botched that, but it's also CCK is what it is for short. And so this piece right here, the duodenum and the CCK, it kind of tells us what's going on and what kind of nutrition is in the duodenum, and it will also limit our next meal size. So all of these different pieces are associated with our regulation of feeding, and basically how much we eat, how we know when we're full, and that kind of thing. So there's also some different factors here related to glucose, insulin, and glucagon. So digestion converts much of a meal into glucose, which, if you may remember from earlier lectures, glucose is one of our main ways that we fuel our brain and other parts of the body. And insulin and glucagon regulate the flow of glucose into the cells. So let's take a look at that feedback system. So our blood glucose levels when they're low, and the insulin levels decrease, glucagon release increases, and some stored supplies are converted to glucose, which enters the blood, slowing the return of hunger. So basically, it kind of starts when our blood sugar is low, we become hungry, we eat, blood glucose increases, insulin release increases, insulin helps glucose enter cells for storage, and hunger decreases. So that's kind of the cycle here, that ideally it's going to be based on our body being aware when our blood glucose is low and trigger that hunger and cue us to eat. But what about when you have high insulin levels? Maybe that's something your body's doing, is you just have these high levels of insulin. Or sometimes people who are figuring out how to manage their diabetes may have taken too much insulin, for example. So when we have too much insulin, it basically shows here that there's a more rapid return to hunger, because these different pieces, the glucose, glucagon, insulin, that they're not really working together in the way that we just saw in the other diagram. So the blood glucose increases, the insulin level is already high, and the insulin helps glucose enter the cells, but the blood glucose levels decline, but not the insulin level. So it kind of stays high, so then the hunger will return more quickly. So someone who maybe has low glucose or low insulin, excuse me, so let's take type 1 diabetes, for example. So basically it's going to look a little different for them too, because there's that hunger and they may eat something and blood glucose increases, but insulin level is low. And so glucose does not enter the cells and leaves in urine and feces instead. So hunger remains high and blood glucose levels stay high, but the cells are starving. So for people who are, maybe struggling with diabetes and don't know it, I know I personally have known a few people that they basically reported one of their initial symptoms was really craving juice. They were trying to get the sugar to try to help their starving cells out, but their insulin just was not, they didn't have high enough insulin to help them nourish their cells. So it's kind of a cycle where they were drinking this juice or craving sugary things to try to get their blood glucose to nourish their cells, but the insulin wasn't there, so they weren't able to get nourished. So the last thing for this lecture is the brain mechanisms associated with hunger. Should be no surprise that, again, you see the hypothalamus is associated with this as well. And these are the different pieces or the different branches related to the hypothalamus: the arcuate nucleus, the paraventricular nucleus, and the lateral hypothalamus. And then we've got these different neurotransmitters that also play a role here. So the arcuate nucleus of the hypothalamus has one set of neurons sensitive to hunger signals and a second set that signals when someone is satiated. So this is kind of telling us when we're hungry, but also when we're satiated and we don't need more. And part of the input to the hunger sensitive cells comes from axons releasing this neurotransmitter ghrelin. So all of these different neurotransmitters and pieces of the hypothalamus, I think they can kind of best be captured in this figure here. And as you can see, there's a lot going on here. Okay, so we've got these different neurotransmitters, and then as we had talked about earlier, we've got other things involved: insulin, glucose, CCK, So I think it would probably be best to really just sort of review this figure and take a look at all of the different complex things that are going on related to hunger and being satiated. So it's kind of a complex process. So I would just suggest go ahead and read through and understand this figure and what these different pieces are doing and how they're all interconnected. And as far as the brain, the different pieces of the hypothalamus that I was just referring to, you can kind of see them here. Here's the hypothalamus in general. And then we've got these also these different areas of the brain that are connected. For instance, the prefrontal cortex can help facilitate food-seeking behaviors. So some of these brain mechanisms related to hunger are a little more complex. And of course, if you have any questions, let me know. But that is about it for what I wanted to cover for internal regulation this week. And I hope you all have a great week. And again, let me know if you have any questions. Thank you so much.